We'll move on to the next speaker. Okay. Yeah. The next speaker, Dr. Chittaranjan Yajnik. He's the professor and the current director, the diabetes unit of the KEM Hospital, Pune, India. He was the Helmut Mahrat Award of the International Diabetes Granted in 2009 and as Outstanding Investigator Award for the World India Diabetes Foundation in 2016 and the 41st Gopalan Oration Award Nutrition Society of the India in 2017 and 18 and has got many awards to his credit. Over to Dr. Chittaranjan Yajnik, please.
Thank you, chairpersons, and thank you, Nirja and Dr. Ganguly for inviting me. I'm going to speak about gut microbiota in diabetes mellitus and also add a little bit of sort of information on microbiome of B12 deficiency. I come from KEM Hospital, Pune, and this is the only diabetes department in the world where the logo is mother and baby because of our intergenerational and now transgenerational work on transmission of diabetes. We couple this work with modern day rapid lifestyle change. You can see here the modern Ganesh, whose lifestyle is very different than what we knew about in the past. I also represent two societies, the SNEHA India, which is Society for Natal Effects on Health of Adults, and International DOHAD Society, Developmental Origins of Health and Disease. With this, we have our efforts at primordial prevention, and the last talk was obviously very important and relevant to these ideas. I have to say I have been helped by Dr. Shrikant Bhutte, my PhD student who did work on diabetes in this presentation. In addition, the slide is not moving. Yeah. And I have to acknowledge help from many colleagues, starting with Yogesh, Saroj, and a whole lot of people with whom I have worked. Our funding has come from Indian agencies as well as agencies from abroad. And we are going to cover today broadly the diabetes microbiome concept, studies which led to it, Indian studies, especially in Pune and Chennai, some trials on probiotics for diabetes from the international literature, and the challenges and opportunities. This, of course, has been said again and again. There are lots of bacteria within and outside us. And there are lots of associations with a lot of conditions. And I think we really need to now sit, take a look, and decide where we need to make a further progress to make it etiological and related to the sort of causality. So the way I see is that in last 10 to 12 years, for a diabetologist like me, who knew about type 1 and type 2 diabetes, both characterized by high glucose, lipid abnormalities, autoimmunity in type 1 diabetes, insulin secretion defects and insulin resistance, and the driver for type 2 diabetes, the adiposity, we have a now new player, that's microbiome dysbiosis. This has been described in various studies, but we are struggling to understand whether it's a cause, consequence, or very likely both. The studies on diabetes and microbiome, there have been human as well as animal studies. And in last 15 years, if you just try to Google on the PubMed, there are more than 4,000 publications related to these. Most of the studies are cross-sectional and retrospective, which makes it difficult to understand causality. There are a number of other comorbidities which might add a confounding factor. There is also an effect of treatment, various supplements, and the pre and postbiotics, either taken as supplement or coming from diet, like the previous speaker suggested. So incisive mechanistic causality studies are still lacking. I'll just show one slide about type 1 diabetes. There is a big international effort. This is really new and we are learning, so it's very preliminary. The study is called TEDDY, it's driven from Sweden. And that has shown actually in type 1 diabetes, lot of population level heterogeneity of microbiome. It's coming mostly from white and non-Hispanic people. But overall impression there is that there is excess of streptococcus and lactococcus in the stool of patients with type 1 diabetes. And it's been linked with reduced butyrate production, reduced bile acid metabolism, and increased LPS. And we heard a brilliant talk by Dr. Bhatt, how these might be related to various suspected and unsuspected metabolic processes. The real hope for a clinician is that if we find a probiotic which might actually change the conversion from the preclinical to clinical type 1 diabetes, that would be a fantastic thing. But it's a dream and it's a long-term dream. There is a review on studies in type 2 diabetes, which summarize 42 studies of association of type 2 diabetes and microbiome. And the resounding message is that several diversity indices and the famous bacteroides to firmicutes ratio has very inconsistent associations with T2D. Let's take one like say alpha diversity. There are 13 reports of like say Shannon type of analysis, no association in 9, positive association in 2, and negative association in 2. If you go one by one, it's the same story for different microbiome sort of components. Though broadly, we can say Bifidobacterium, Bacteroides, Faecalibacterium, Akkermansia, and Roseburia are negatively associated, while these Ruminococcus, Fusobacterium, and Glaucia are positively related. Lactobacillus seems to be of great interest, especially in India, but the associations are inconsistent across the sites. And that is shown pictorially here for the different, like say, phyla or whatever you call it. Here they have a, code that negative studies are shown in blue, positive in red, and you can see for the same bacteria, there are sometimes both types of associations. In other studies and other population, it may be only one-sided, and in animals, it might be very different. Overall, to say that this phylogenetic tree and its components have a very different association in different studies, and therefore it's not possible to make an overwhelming positive or negative statement. What I really feel the message there is that we need population-specific markers if we are to actually make a progress in this. And this, of course, takes us in the realm of what is a buzzword today, either precision medicine or personalized medicine. And towards the end of the talk, we might see what might be the future designs of the study. But this should be a central issue that the patients and the controls should come from the same population. They should be matched in other confounding factors. Then only you are going to be able to make some sense of these associations. If we now look at Indian diabetic patients, like the previous speaker said, Indians have a heightened genetic predisposition, but more important than that, and especially for research which has happened in my department over the last 40 years, we have shown a resounding role for intrauterine undernutrition and subsequent relative overnutrition, so-called mismatch hypothesis, which is a driver of epigenetic programming of diabetes in Indians. This leads to an earlier age at diagnosis at a much lower BMI. The environmental factors which precipitate it are rapid economic transition, rural to urban migration, and now we have the very dubious distinction of being the world's capital of diabetes with estimated more than 100 million patients in India. Indians are different for microbiome, that is these biases, as well as the phenotype. And my department has contributed quite a lot to definition of phenotype of Indian diabetic patients. As I said, there is genetic diversity, there is endogamy, there are diverse dietary habits and geographic locations, even for differences within India. And I actually described the Indian phenotype, which we called thin-fat Indian phenotype or the thrifty phenotype, where we showed that for a low BMI, we have higher body fat percent, we are more centrally obese and more dysmetabolic compared to the Europeans. And 40 years ago, this was a one-man, one-control study, myself and my registrar in Oxford. And in one study, we could find major differences in all these factors. For the microbiome, the group I work with, Yogesh Shauche's group, written by Shrikant Bhute, there are clear differences at the family level distribution of the bacterial taxa. If we compare, like, say, Indian, Bangladeshi, American, Spanish, and Korean, with a suggestion that within the Indian subcontinent, there are more similarities compared to the other parts of the world. If we were to look at the overlap of OTUs at 97% similarity, only 3 OTUs are common to all these populations in the studies of type 2 diabetes. And if we look at distinct OTUs for other populations, they are to the tune of 100 to 200. For Indians, they are more than 450. So we have a much more diverse microbiome than the other populations. If you do the principal component, then Indians, of course, stand out very different than the Americans. And if you look at patients and controls, what this slide shows is that Indians have even more diversity within our groups also. So that adds to the complexity of associating the microbiome with the phenotype. And we need a consortium and very specialized studies with a lot of thinking to make sense out of this. What are the studies from India on microbiome? 2 studies from Pune. The first one from my department, Shrikant Bhute. Then again, his sort of department with Gaike as the first author. And 2 studies in an Indo-Swedish collaboration between Dr. Mohan's unit in Chennai and Copenhagen. Their numbers are bigger because we had very small grants at that time. And the specialty of our study was that the samples came from one of the 3 prospective birth cohort biobank that I have. So we had ready-made samples in biobank, which were serially collected over last 30 years. And in the Pune children study, in the parents of the children, we were able to find 19 healthy subjects, 14 newly diagnosed diabetes subjects, and remaining known diabetic patients. This allowed us to have actually their extensive phenotype over years and a sample of stool whereby we could do the microbiome. And Shrikant's special approach to this under Yogesh guidance was that we looked not only at bacterial diversity, but we also had special primers for archaea, fungal, and eukaryotic microbiome. And that actually makes it very distinct from other studies because we were able to find differences across the board. And what were the findings? We had a reduction in alpha diversity in patients with high glucose, whether newly diagnosed or established, compared to the normal glucose tolerant people. If we look at microbial dysbiosis, there were a number of phyla like Bacteroides, Firmicutes, Proteobacteria, Actino, etc., where there were differences between the populations. If we look at mean abundance, 20 discriminative OTUs showed a characteristic transition from the normal glucose tolerant to early or newly diagnosed patients to the late or already diagnosed patients, suggesting that as the metabolic abnormality increases, the differences become more pronounced. And then we actually look at differences of, like, say, total bacteria. They were much less in the diabetic patients compared to the NGTs. Similarly for Bacteroides, but for Lactobacillus, it was exactly opposite, that the diversity, or sorry, the absolute count was much more for Lactobacillus in the diabetic patients compared to the other two groups. Again, showing how complex it is. The principal components again showed the three groups stood out different, but on further analysis, we find that there is actually diversity across the group, which is not restored after treatment of diabetes. In other words, it continues to remain with the patient. And this is a cartoon which Dr. Bhutte drew, which summarizes all these findings. And for me, I have to just summarize that actually our impression was that the microbiome dysbiosis which we were observing could be a consequence rather than cause of the high glucose. And this was because, like say, with higher glucose, there were more disturbances. But equally, we found some evidence that as a dysbiotic state is reached, it itself affects the gut wall integrity, causes endotoxemia and chronic inflammation, and exaggerates the two basic deficiency or basic defects in diabetes, namely insulin resistance and deficiency. So then it becomes a self-propagating sort of phenomenon. If we look at other study from Pune, where they added one more group of pre-diabetes to the normal and the two groups of diabetes which we described, the findings are broadly similar. And here, the pre-diabetes and NGT are closer to each other than they are with the higher glucose, again suggesting a dose response type of phenomenon. This study additionally measured circulating metabolome and showed very interesting findings relation to different metabolic sort of domains like oxidant stress, inflammation, amino acid metabolism. But interesting to show that the different components of microbiome have very different associations with the same molecule in the blood. And therefore, I think this provides a rich database for future investigation to see which of these components can be picked out.
In 2 minutes?
Maybe a little bit more. I had negotiated with Dr. Nirja because she asked me to give B12 also.
Oh, okay. Sure.
Sorry. So in other words, we found a lot of similarity between two Pune studies, and that actually could be a geographical thing. Then comes the studies from Chennai, which are in collaboration with the Danish group. Here we have two groups from India and two groups from Danish. normal glucose tolerant, and pre-diabetes. And very interesting impression you get by looking at this heat map is that there are more differences between the countries than between the two metabolic groups. And this is again one more indication that there is great diversity across geographical areas, and therefore we need strong local controls to make sense out of it. And same thing at taxonomy level and at function level. Therefore, what they said was they found 16 OTUs which were enriched in normoglycemic and 144 in pre-diabetes. They were actually separately additional gut microbiome signatures when pre-diabetes was discovered. Several Prevotella OTUs were enriched in pre-diabetic while Megasphaera in IGT. But interestingly, these are very much enriched even in healthy Indian individuals. The pro-diabetic patients had higher pro-inflammatory state, and these were different than the Pune studies, again highlighting the differences. Then second paper from that group reports in type 2 diabetes. And what interested me here was that they extend the findings, but they say that T2D related microbial changes at taxonomic level were seen only in the Danish population, not in India. So again, more food for thought. Things are getting quite complicated. One of the reasons which I have actually for studying serial birth cohorts is that we have demonstrated that metabolic abnormalities in adult age can be tracked back to very early age, like 6 years of age. Here, those who are pre-diabetic at 18 years were already pre-diabetic at 12 and 6 years. And they already had metabolic abnormalities which lead to it. In other words, if you study only adults, you get only an end-stage cross-sectional picture. We really need to go into early life. Now, what about interventions? This is a meta-analysis reported again by Indian workers, Dr. Paul and his colleagues, where they had 58 studies with more than 3,500 people which took part. The studies were extremely heterogeneous, but there was a common finding in the meta-regression that with with the probiotic treatment, there was a fall in fasting plasma glucose, HbA1c, fasting insulin, and HOMA-insulin resistance. This tells us that it might be very useful, of course, to follow this, but we have to be very aware of the confounding factors like age, BMI, baseline markers, dosage, intervention duration, and the study region. But the hope is that there is hope. So I now think that we are moving around in a vicious circle. We don't know which comes first, chicken or eggs. So there is dysmetabolism, dysbiosis, which drives the whole cycle of exaggerated inflammation, dysmetabolism, complication, and adverse outcomes. And where to enter this cycle would be interesting investigation. We have published genetics of subgroups of diabetes in India. And what I would like to point your attention is with type 2 diabetes and its subtype, the moderately obese subtype in Indians, but not in the Swedish, giving us some indication that B12 might be involved. You know, 30 years I have worked on B12 and folate and found a lot of influence on fetal programming and programming of diabetes. So this was a very rewarding research. And then we did B12 deficiency microbiome studies. Lots of collaborators and students, but especially colleagues from UK, from University of Kent and Quadram Institute. And what we find is that there is a variance of microbiome in the B12 deficient and B12 sufficient people but it's also substantially influenced by the age of the person. So that in children, that's boys and girls, and fathers and mothers, the differences are more than within the B12 deficient and sufficient groups. Then we looked at the phyla differences and found that there are differences in firmicutes, actinobacteria, proteobacteria, and Bacteroides and cyanobacteria. And if we go to the next level, then we can see quite distinct differences in relative abundance of a number of genuses. And to me, this is the first demonstration from within India that the genera of the microbiome associated with B12 deficiency are different. They are influenced by the age of the patient, sex of the patient, and also the vitamin B12 status. Some of these different bacteria are actually producers of B12, while others are consumers of B12. And the hope of this analysis, which we are of course continuing to make it more refined, is that if we are going to be able to improve the population's B12 status, we will have to create probiotics with more of B12 producers and less of B12 consumers. Is there any other evidence for this? And I'm thankful to my colleagues from Tata Consultancy Services, Krishna, and I think Pina, who is attending this meeting. He has shown this slide, I think, in his presentation, that they did a sort of a meta-analysis of microbiome in different populations, related it with the average BS in these relations as available from the literature. And what they find is mean abundance of vitamin B12 producing bacteria decreases as the B12 deficiency increases, while the B12 consumers increase as the deficiency increases. So it must be a dysbiosis between producers and consumers that might reflect at least part of the B12 status of the host. So for future, I think we need much larger number of participants in our study. We need and locals. We need prospective rather than cross-sectional studies. We need functional microbiome analysis, including ultra-deep metagenomic and preferably metatranscriptomic sequencing, combined with metabolomic assessments, not only of the bacteria, but also of the host. And for host, we need genetics, lifestyle, and multiomics. I am actually trained in epidemiology, where Mendelian randomization is now assuming great significance in trying to prove causality in different studies, in addition to human intervention, which has its own limitations because of the cost involved, the designs involved, and a whole lot of things. So I think we should think about Mendelian randomization. And ultimate hope will be to have clinically translatable products. These ideas were told to us by an economist. Many of you perhaps know. This is my last slide.
Professor Yajnik.
Yeah, this is the last slide, that we need to keep Bradford Hill's octet in mind for future studies in the microbiome. Thank you very much.
Thank you, Professor Yajnik. Sorry to interrupt you in a couple of times because the program had only 20 minutes for you.
Thank you.
Okay. And now we will have the question answer session. So I request Adra to come on stage. Sabu and I will coordinate this. So the questions are for all the three speakers of this session, and I would ask the audience offline first to ask the questions.
I have a question for Dr. Ardra, so it's an interesting model that you talked about with the notobiotic mice showing changes with the glucuronidase. Now, the thing with the cancer drugs is that some of them affect the small bowel and some of them affect the large bowel. Checkpoint inhibitors, for instance, would cause colitis. The irinotecan that you talked about and maybe mycophenolate would cause small bowel diarrhea. The question really is it bacteria in the duodenum which produce beta-glucuronidase which are important, or are they colonic beta-glucuronidase producers which are important?
Thank you for your question. That's very astute of you. We have yet to undertake a specific and systematic biogeographical examination of beta-glucuronidases in the gut. I do think that their overall intrahepatic circulation, recirculation probably contributes to some of duodenal, small intestinal damage as well as colonic damage. So yeah, I don't really have a clear answer for you.
Actually, Ardra, we need to have this answer because as Dr. Yajnik was saying, that we need to intervene when they're 5 to 6 year old children. And TAMIS and KEM is doing a study where they are moving with intervention at this stage. So a lot of misery could be. So emanative, sudanitive, dasanitive, when they are given for the chronic myeloid leukemia, they move as a whenever there is a blast formation, you move the new drug and then you move the new drug. So if we could intervene in the gas sector and with ICANN or whatever you have and identify them early while they are being treated, we can perhaps avoid use of the consequent drugs.
Absolutely.
So there's for both Dr. Yajnik and you that the best method will be to intervene them at a period when you could do something, because if you look at the Yajnik study, they even when they have been treated for diabetes and controlled it, the microbiome is remaining same. It is not changing, and the metabolomes are also same. So if you intervene, you have to continuously intervene to them, which is very, very difficult. So why not intervene very early into this? And to two very small questions. One to Dr. Yajnik, that you have followed these girls for nearly 40 years. And if you have the data, have they changed the data? Because your data showed at one time that if the epigenetic signature didn't change, so they went on producing the same challenged babies. So does the microbiome changes in these years in them, or it remains same? And Dr. Sanjay Banerjee, I have a question for you that this is very interesting. But if you are looking at one of the species of the lactobacillus, Baustosh has got a huge number of signatures of the same species. So you need to really find out more about these signatures, these sequences which you have, and you have chosen NGT6 or 26. I've forgotten the number at the moment. But if you choose these signatures, which are very important signatures to look at the metabolome and other changes which are happening there, the study will become richer. Thank you.
So we'll start. You wanted to, no, okay. Yeah, Dr. Yajnik.
Okay, thank you, Professor Ganguly. As you know, we conducted world's first primordial trial in adolescents in our cohort to improve the health of the next generation. So that's even before the childhood. And that is an intergenerational influence. And we already have results of the neurocognitive improvement in the children by providing only 2 micrograms of B12 to the adolescent mothers from the time of adolescence till the day of the delivery. We are now following up these children between 6 to 10 years of age to see if we have changed the cardiometabolic risk. And that study should be over probably next year. So I'll be able to tell you more. What we have observed is that the children born in the trial have about 250 grams higher birth weight compared to their mothers. They have larger head circumference, they have larger abdominal circumference, but very interestingly, they do not have excess adiposity. So I think whatever we have done over years by way of general improvement in the background due to advance or development in the nation, plus our intervention seems to help us to come out of the thin fat phenotype. The definite answer will come a little later. My microbial biobank started only about 10 years ago, because before that we had no idea about this. So we have blood samples. So if we can find microbial signatures in the plasma and serum, and I know there are, then we can go back 2 generations and find whether it has changed. And I seek your support in obtaining funding for this. Okay.
Banerjee,
Thank you, sir.
Banerjee, Dr. Banerjee.
Yes, thanks, Dr. Ganguly, regarding your interest. See, I'm also very new in these topics, and after coming to Assam, I realized that in Northeast they use a huge amount of fermented food, and then my research started, exploring them. So yes, one of the species we have started working on it, and we don't know that actually whether this is different than other similar species from other strain. So I sent the bacteria to Dr. Bhavata's lab to go to the whole genome study to look at how they are different from others. That is important. What I said, we are going it when you do this. Also, we send the fermented food to Bhavata's lab to do the metagenomic study. Whatever we have isolated, it is still present into the bamboo shoots or not. So he'll isolate the bacteria, whatever present in the bamboo shoots, he can look the presence. So which will give me confirmation whether whatever we isolated, that is also present. And whatever you said, very interesting that we should look the little bit on metabolomics point of view. If after 14 weeks, we really see there's a reduction of insulin resistance, and also I can see there's a tendency to reduce body weight, which was previously reported that adiposity, anti-adiposity effect is there with the lactobacillus brevis. So if it's there, I think it's interesting that see that how we can, take it, more forward, go for more, study. Like what is the probiotic property? There are some ICMR guidelines, but that guideline is, very, it is not that they've done everything. They're having a 5 and 6 some property they want to do in vitro. But if you really think that is a very good effect preclinical, I'll come to you, sir, and I need a discussion what to do, what to take it forward. So, and I'm really excited in the field, and a lot of hope is there. So thank you very much for giving me the opportunity.
Dr. Sanjay, can I suggest one more thing, measurement in your samples?
Yes, sir.
Because vitamin B12 is made only by microbes in nature.
Yes, yes.
And if you also measure B12 and 1-carbon metabolites in your brew, I think that might give entirely additional viewpoint on what might be actually useful in another way. Because B12 deficiency is quite rare in the Northeast.
Yes.
Okay.
So, sir, I think after your lecture, I realized actually, that vitamin, B12's, source basically, a lot of different bacteria which consume, and some of them are producing. So those are very important things to look into it. And I think you might know that in Northeast, I know how much microbiome work is done, and our Northeast signature will be completely different than other part of India. And here cardiovascular disease is not that big, but overall, cardiometabolic diseases are there, but they are somewhat protective from, as I said, Nagaland, Meghalaya, they having a less cardiovascular disease compared to other states. So we really need to explore actually this area, how they are different.
I have set up a birth cohort and an adult cohort in the Dibrugarh Assam Medical College. It is
Yes, sir.
biobank.
Okay, sir, someday I have
And
to
you
discuss
can
more.
benefit.
Dr. Bhatt, this is based on your presentation, I have a concern.
Yes.
And it is, see, when we do phase 3 drug clinical trials in different ethnic groups and different populations, we are factoring in the variation that exists in the drug metabolizing cytochrome P450 kind of enzymes. But we never ever thought about the microbiome altering the safety or even the toxicity of the drugs across population. Now, this is a big issue, especially when you raised on the anti-cancer drugs, and we never studied this at all.
Thank you so much for preaching to the choir. Thank you for bringing that up. That's definitely the reason why we're doing this work, because the importance of bacterial drug metabolism is largely understudied. This is a very new and developing field. And like you're saying, you don't see what this does until you actually do the work in people. And at that point, we've spent $1 billion on developing a compound that ultimately ends up in the trash, right? Because of too many side effects. So this is why I think this work is incredibly important, because we've developed, mathematical models to estimate how much drug is actually reactivated in the gut by bacteria. For just one enzyme, beta-glucuronidase, there's millions of other enzymes that we don't even know about. Well, maybe not millions, but thousands at least, that might be involved in metabolizing drug intermediates. For example, glucuronidation is not the only method of detoxification, right? There's also sulfation. So sulfatases are, bacterial sulfatases are a largely understudied group of enzymes. So yeah, you're absolutely right. This is a critically, this has been an overlooked aspect of drug development and drug metabolism. And thank you so much for highlighting the importance of what we're doing. I appreciate it.
Yeah, Dr. Adra,
Can I say one thing in diabetes, metformin is known to affect microbiome, and microbiome is known to affect metformin metabolism and efficacy. So that has been known for some time.
Dr. Adra, nice talk. Just curious, is there any evidence to know how quickly after birth are these bacterial glucuronidase expressed?
Wow. I've actually never thought about that.
Yeah.
But we do know that a number of lactobacilli express beta-glucuronidases. I mean, the availability of so many birth cohorts would be a great way to actually examine the development of what we call the GUS-ome, or the beta-glucuronidase ome. We call it the GUS-ome. I think those will be really great. We have fantastic bioinformatic pipelines to actually quickly determine GUSs based on actual authentic structural data that we have. So that would be a great thing to do.
Yeah, the only reason I ask is because in conditions like kernicterus, where it's supposed to be a problem with the beta-glucuronidase not being active at birth, and having to treat it so that it is activated and the bile is conjugated and excreted out. So is the problem that beta-glucuronidase is not active, or the bacterial glucuronidase is overactive?
What was the disorder you spoke of?
It's called kernicterus. It is
right. So I moved rather fast, but so there's very specific structural features that we've found that contribute to substrate specificity amongst bacterial GUSs. that particular structural feature is absent in the mammalian homologue of gusses. So because in biochemistry and in biology, form fits function, there's a very distinct substrate preference for bacterial gusses versus mammalian gusses. So from what I know about the story, it seems like the mammalian gusses are more important for the bilirubin recycling and not bacterial gusses. Bacterial gusses, like I've showed you, they either work on small molecule glucuronides or endobiotic glucuronides, such as certain hormones and neurotransmitters, or on really large molecules like heparins and certain large glycosaminoglycans. So,
Dr. Bhatt, can I offer you a collaboration? We have serial samples on the stool samples from the children born in our third generation, starting with meconium and every 3 months for first 2 years along with mother sample.
I look forward to chatting with you offline.
Okay,
That sounds
thanks.
amazing. Thank you.
Agra, that was a lovely talk and
Thank you.
it's a lot of good research. I was only thinking about the idea that whether the point would drive the point home that usage of probiotics would be undermined. It would be less in use, where we are trying very hard to push probiotics in the larger interest, global interest, for betterment. So it was just a thought because we have not researched on that topic at all.
I propose my work as a complementary approach to using probiotics or live biotherapeutics or symbiotics or things like that. This is a complementary approach to that, which I believe, which I agree is invaluable for improving health.
Yeah, I think there is no more questions from the audience, but we have a few questions from the chat box also. So we are running out of the time. I think we'll go ahead with the chat box questions very fast. The question is from Professor Prakash Halami, and the first question is to Dr. Sanjay Banerjee. Does the consumption of the fermented food, such as the bamboo shoot, is credited with a lower risk of cardiometabolic disease? Is there any specific evidence available for this? Dr. Sanjay Banerjee.
Yes. So there is no specific evidence till now. These are all belief on through traditional knowledge that whoever consume traditional food like this kind of bamboo shoot every day, so they having a better health. But there are actually some identification of bacteria from this bamboo shoot are done, and they suggest that because they have a good lactobacillus bacteria and that might be benefit, but this is an indirect kind of evidence, not a direct evidence. So we are trying to do that. We want to do the preclinical study where we're giving the bamboo shoot itself to the animal, as well as that probiotic, all those bacteria we have isolated we can give, and parallel see that possible is that bamboo shoot itself has a lot of good metabolites or flavon or all those present fiber. Those also may be beneficial effect. So you have to do some study, then only I can answer these questions.
Okay. Then we will move on to the next question from Dr. Vimala. The question is to Professor Chittaranjan. The question, what is the impact of the diabetes medication on the microbial dysbiosis?
Okay. I mean, metformin was the first drug where it was shown to actually affect the gut microbiome of the patient. And there is a lot of literature on this. Other drugs which might be acting, I don't think they have been studied systematically. But for metformin, there is a big story.
Yeah, we will have one more question that is to the Dr. Meera, to Dr. Adra, but my question, the question is do you believe there is uniformity in the bacterial population in the cancer patients you studied?
Wonderful question and very astute. No, I think people's microbiomes differ, and they particularly differ based on your diet, geography, as a lot of the work in the last 2 days has shown. However, what's really interesting about bacterial beta-glucuronidase that I don't have a lot of time to talk to you about in detail is that it's an enzyme whose expression is subject to catabolite repression, which means that this enzyme is expressed by bacteria, the Enterobacteriaceae in particular. It's upregulated up to 5,000-fold upon the presence of a glucuronide substrate. So in work that we've done so far, it's pretty evident that the presence of a medication that is glucuronidated within, a mouse model, for instance, does upregulate the expression of beta-glucuronidases. So again, this allows us to develop a targetable and a druggable enzyme and function.
Okay, thank you. As we are running out of the time, we have already answered so many questions from the chat box, but we don't have enough time to answer all this. So this will be answered through the online mode very shortly. So I think we will stop this session here. Thank you very much. Thank you all. All the participants are sitting here in the audience and as well as in the online mode. I also take this opportunity to thank the organizers for putting one such mega event in the southern part of the country, in the state of Kerala, in the extreme south of India. Thank you very much, especially by hosting the RGCB Director, Professor Chandrabas Narayana. This is very important event that is happening for the first time in the state, especially in the context of the initiation of a new dedicated center, the Center of Excellence in Microbiome in the state. That is what we are just mentioned is, in association with the Rajiv Gandhi Center for Biotechnology. So thank you very much. Thank you, organizers. And also for giving me an opportunity to co-chair this fantastic meeting. Thank you. Thank you all.
Thank you, Sabu. I think he's going to let us felicitate, right? No, your mic is on.
Sure. So thank you so much. So now it's time for the felicitation. So I request the chairpersons to felicitate Dr. Adra Bhatt. So thank you so much, Dr. Adra Bhatt. So I request to take a snap together. Yeah. So now it's time to felicitate our chairpersons. For that, I would like to welcome Dr. Siad and Dr. Rajesh onto the dais. You also will get one now. So thank you so much, Dr. Sabu Thomas. Thank you so much, Professor Chandrabas Narayana. Thank you all. Thank you, Dr. Siad and Dr. Rajesh.